So welcome back. And today we get to talk about plant breeding or hybridizing and understand the importance of it as it relates to plant propagation and developing new crops. And also to understand that plants may have different type of mechanisms that is going to prevent them from taking the pollen from a different species. They don't want to breed with another plant. They just want to breed with somebody from their own species. So apples with apples and oranges with oranges. And so we have the transfer of the pollen, which is going to continue what we already started as far as the sexual reproduction and the usage of the usage of the flower. And so when we have the reproduction of plants, there's going to be certain things that is going to be important for you to know. For example, parthenocarpic. Parthenocarpic is going to entail plants, certain plants, not all of them, that are going to produce the fruit without having to go through the process of pollination and fertilization, which in most cases then may be sterile. And so the case is, example is going to be like plain simple banana fruit. Banana fruits are going to be a hybrid, and their seeds will never develop because the alleles or the genetic material is not going to pair evenly or equally. And so it's always going to have the embryos that will abort, and thus the plant still carries on the production of the fruit as if it has been fertilized and pollinated. So it's kind of like a phantom pregnancy if we were to compare it to humans, where the plant thinks it's going to be producing the seeds, but in reality, they will not. And so certain plants do provide or have this ability of creating the fruit without having to go through the process of pollination fertilization, and a few other examples, including the figs and the cucumbers and a few other uh, plants that will do that. And so we have the transfer of the pollen. And this is very important that plants like to have the pollen from a different individual. They do not like to self because selfing causes what is known as inbreeding. And so plants naturally or normally would like to have the pollen from a different plant of the same species that will bring about genetic diversity. And having genetic diversity is going to be important for plants in case there's some kind of catastrophe, then hopefully one of those two plants will survive. And so we have here the example of corn, where we have the tassels that are going to be the male flowers, and then we have the silk, which are going to be the female flowers. And so the pollen from a tassel has to land at the tip of all the silk to bring about the pollination and then fertilization. And eventually you're going to have the corn kernels. So the transfer of pollen is going to be very important. Plants want to make sure that their offsprings are going to be genetically diverse. And that is why it is going to be difficult or impossible to take a seed from a golden delicious apple and you grow the seed and you're not going to have a golden delicious apple. You're going to have something else because it came through the process of sexual reproduction, which means that genetically is going to be different. And so here's some of the terms that we're going to go over. Outbreeding, when plants want to have the pollen from a different individual. Inbreeding, where plants can self. Many plants will be able to self. As we have selected plants and we hybridize them, we might also take out whatever mechanism they have in order for them to prevent them from inbreeding. And so we have purposely for our own use have removed some of these mechanisms from the plants. Sexual reproduction, which involves the flower, the ovule, and then asexual will continue on the next time with the asexual or the cutting propagation of plants. So very important to know this. And so a lot of this work started with Gregor Mendel, the Australian I guess geneticist will be now the term used, when he started just making observations and doing deliberate crosses of different types of pea plants. And so the Punnett square, as we see here, I'm sure you probably saw it in your high school biology, where he crossed some of the pea that were purple flower and with a white flower, and then created some of them that would have all purple. And so what we happen here is when you take two individuals and you cross them, then you get the philia or the first philia, which is the F1. You may have seen some of this in some of the seed packets. So that is the progeny of the first cross. And so what Gregor Mendel found is that plants have traits, like in this case, color, and some of them are going to be dominant. And so the first generation, which is going to be the F1, all going to have the dominant color, which in this case is going to be the purple or this lavender color. But then taking the offsprings or the F1 generation and cross them, then you're going to have some of the recessive traits. And in this case, the recessive trait happens to be back to the white. And so that was the beginning of the genetic work that eventually has led to all the genomes that we have now, and eventually has led to the genetic engineering and all of that. So it had to start somewhere, and that was with some of the early work by Gregor Mendel here. He not only crossed peas and studied the color, but also the shape and the color of the fruit, because all of those are determined by the genetics. And so in most cases, there is going to be a trait that is going to be dominant, and that's going to be expressed by all the individuals in the first generation. And then as time went by, there's the work with genetics have kept on getting better and better. And so certain plants were found to have zero dominance. So they have no genetics that is dominant. So when you take, for example, a snapdragon, and you take the red and cross it with a white, then your first generation is going to have all pink. So no, not red like the previous one. And then when you take the F1 generations and cross them, then your F2, your second generation, is going to have an assortment of different colors. So certain plants decided not to have the genetic trait that is going to be dominant. And then we have some pinks or carnations that are exhibit something similar, where there's an absence of a dominant trait for color. So in order for us to cross plants, we need to have both male and female component. And so very, very often, you're going to have plants that are going to be considered perfect flowers. And that is because they have the male and the female component on the same flower. And so here is the Aztec lily. And when we look very closely at it, we can see the stamens that have the pollen and the stigma or the female component. So it is very important for it to have both of them. And so one of the first type of crossing is going to be the outbreeding, where the pollen from a different individual, a different plant, is going to be delivered to the stigma of a second plant. And so this is what many plants will do. And plants have taken steps into ensuring that they get the pollen from a different individual. So here, just to delineate, we have the parent plant as a double capital A, and the second parent plant, or the pollen donor versus the egg donor, as the lower caps, just so that you can see. And so when they cross, or when the pollen from parent 1 goes to parent 2, then the offspring is going to have the A capital and the B A lower caps as well. So it's a combination of both parents. And so plants had to figure out ways of preventing the same pollen or the pollen from the cell, the same flower, to go into the stigma, because that's what's going to bring the inbreeding. And so many flowers opt for maturing their sexual organs at different stages. So for example, when we have the protogyny, This means that the flower, the female component, which is the gynecium or the carpal uh, pistil, is going to mature or be receptive before the stamens can drop the pollen. And so that is going to ensure that the flower is going to receive the pollen from a different flowers. And it could be a span of several hours or sometimes a span of a day. So a flower can be female receptive for one day and then close, and then the next day is just going to be male receptive. And so by having the stigma or the female component mature and being receptive ahead of the stamens and the pollen, then that ensures that the pollen from a different plant reaches the stigma. And so we have the example more common of the plant known as the calla lily. And so we see on the left side, on the first day, we have the female flowers that are going to be towards the bottom that are going to be receptive. They're going to take the pollen during this stage. And on the second day, the female components will be turned off, so no longer accepting the pollen. Now the male flowers that are going to be the top portion of this column is going to release the pollen, and the pollen will go to a different flower. So that ensures that it's not going to self or prevent the selfing. And then we have the opposite, which is protandry, sorry, protandry, which means the andrecium or the stamens are going to mature first. They're going to release the pollen. The pollen is then going to be carried to a different flower. So this, once again, could be a span of a few hours or several days or 1 or 2 days. And so here we have a plant by the name of balloon flower. And so right now on the first day, we see the anthers have shed their pollen. They're in the middle of the flower. So a bee visiting this flower will take the pollen and carry it somewhere else. And then during the second day, now we see that the stamens that have done their job, they're now withered and they have gone away. And now we have the stigma that has opened. And if we look from the previous picture, the stigma is inside, but it's not receptive, so it's not open. Second day, now the stigma is open, is receptive. Now it's going to take the pollen from a different flower, and that's going to prevent it from selfing. So protandry is when the stamens mature before the stigma. So that's going to prevent it from selfing once again. And then we have the extreme where plants ensure that there is no selfing by having either male or female plants. So this is extreme where a plant is only going to be considered a male because it's only going to produce pollen, and the female plant will have only female flowers. Now, it is very important that they are in close proximity, but this will ensure that a plant will never self because it's just unable to produce pollen. It's just going to produce one or the other component. And we have here some examples, as in the case of some willows, that they are going to produce the male flowers, and then the different willows going to produce the female flowers. And or here's another example more common. This is carob tree. And on the left, we have the male flowers that come from a male tree. And all we see is a very simplistic flower with just a bunch of stamens. The stamens will release the pollen. And on a very different tree, we have the female flowers. And very simplistic, it's just the receptacle with a very large stigma. And so the pollen from the male tree has to reach the stigma of the female tree. And so only the females will bear fruit, and that is because they have the female flower. The male is just going to produce the pollen, and that's it. They will never have a fruit. So this ensures that there will definitely be an outcrossing and avoiding that selfing. And we have a few other plants like in the sumacs, where we have the remnants of some of the reproductive parts, but no longer functional. So here we have the female flowers with the stigma, and then here we have the male flowers with the stamens. But if we look very closely, there are remnants of the stamens here. So perhaps the ancestor had both functional male and female components, but as time went by, then just adopted one or the other to ensure the outcrossing. And then we also have the example of plants that are monoecious, and that means that they are going to have either male or female flowers on the same plant. Now, the other one, dioecious, meaning you have male and female plants. But these are the same plant that will have either a male or a female flowers, but they're still on the same plant, maybe in different nodes, maybe in different areas, maybe on different times of the year. So they will have male and female flowers. And the example here is going to be our walnuts. So here we have the male flowers that are producing the pollen, and somewhere hidden would be the female flowers with the stigma and the ovary that will later become the fruit. So walnuts, acorns are going to be common examples of this, and or also the members of the cucumber family. So here we have just to common gourd, which the female flowers has the ovary on the underside of the flower, and the male flowers have no ovary. So there's just going to be stamens with pollen. And so they're going to be produced in different areas of the plant. And normally on a cucumber or a squash or zucchini, there is going to be a lot more male flowers that are going to produce versus the female. And so the male flowers are going to be numerous, whereas female maybe 3, 4 per plant. And even here, begonias, we have just same flower cluster with 2 different flowers. So we have the male flowers, and on the other side, there's no ovary. There's nothing here, just a bunch of stamens and the petals. And then the female flowers will have this ring, which is a margin of the ovary, and just stigmas versus stamens. So they're side by side on the same plant, but different sexes on the flowers. And bananas, more common. The first grouping of flowers that will come out of the spike of banana will be female, and you can see them with a much larger stigma. And then at a certain point, the sexes of the flowers will change, and then the plant will produce only male flowers. And if we look at a close-up of them when they are probably receptive, you can see here's the female flowers with a very large stigma, and then those are followed by a male flower that have a much smaller stigma compared to the females in the background. But now they should have that pollen, keeping in mind that they're hybrids, so this is not necessary for them. But you can see the difference between the male and the female flowers. And so we have here some of the ways that plants prevent the wrong pollen or the same pollen from fertilizing itself. So there are genetic barriers found within the stigma. So first of all, the stigma is the first barrier. If the stigma is unable to recognize the pollen, so if it's an apple and the stigma does not recognize it as an apple pollen, then nothing is going to happen. The pollen is going to germinate, but it's not going to have the enzymes to digest the stigma and the style and allow the sperm to go and meet the ovule. So that is going to be the first one. And so here we're using simple letters just to signify. So this is plant that has an AB capital for the stigma. And so it knows that an AB capital for the pollen is probably going to be the self. It's going to be the same from the same flower. So it's not going to accept it. And so here's now example B, where flower is BC. So it's able to accept pollen A because it knows that pollen A comes from a different individual. And so pollen A is going to have the enzymes and is going to be allowed to germinate and fertilize the eggs. And vice versa, when there's a CD, then it's going to accept both A and B. So these are just examples. So what's important is that there are genetic barriers within the stigma that prevents the wrong pollen or the pollen from the same flower to fertilize the eggs. That's what plants do not want. Because then it's going to lead to inbreeding, and inbreeding is going to eventually cause plants to be weaker, and that's what they don't want. Now, inbreeding is good for us because we can keep on repeating the same cross, and does bring about the same plant. So we have almost done a lot of inbreeding on plants that we are creating them almost genetically identical, and or we can repeat it over and over and over again. And so inbreeding is when we're going to allow the pollen to pollinate and fertilize the stigma from the same plant. And as I mentioned before, we have taken some of these genetic barriers out in the process because we want to make it easy for ourselves. So pollen from the stamen is just going to go on the stigma, and many plants will do it naturally. So often, if they'll prefer to have the pollen from a different individual being outcrossed, but if there's an absence of insects, if there's no pollen that reaches the stigma in the time period when it's receptive, then as a last resort, many plants will pollinate themselves with their own pollen. You're still going to get some seeds. You're still going to get slightly diverse individual, but not as diverse as if it comes from a different parent. So keep that in mind that some plants will do it as a last resort. But we do a lot of inbreeding, we do a lot of hybridization. And so many plants, like members of the pea family, you have the female and the male component kind of hidden, tucked away next to each other. So it's going to be almost impossible for them to not pollinate each other or not to inbreed. And so some plants opted for doing this. In this case, all the reproductive components are found in here, and here's the cross section of that drawing. So the plant will prefer for a bee to open the flower, bring the pollen from a different individual, but in the absence of that, or if it's too difficult, then it will still produce a fruit, and that's why you would always get peas when you plant a pea plant. Even though you might have one, you're still going to get peas because it's not going to require the pollen from a different individual. Another important vocabulary word that you need to know is apomixis. And this would be certain plants that have bypassed the pollination and fertilization. Once the flower is produced and all the ovules in the flower are going to turn into seeds, regardless if they get pollinated, regardless if they get fertilized. And so this would be the case of the sunflowers, not the sunflowers, but the dandelions. And there's a reason why dandelions are kind of prolific, because every single time there's a flower, that flower is going to turn into a lot of seeds. And so they don't rely on the insects, they don't rely on weather, temperature, or anything else. They have a flower, the ovules in the flowers are going to turn into a seed. So they just morph into an embryo, and the embryo will behave as if it's a pollinated seed. And the raspberries would be another example where you have a flower, the fruitlets are going to develop and mature as if they were pollinated and fertilized. So that is what apomixis means, a ovule that would turn into a seed, or a ability of a plant to produce the seeds without any kind of pollination or fertilization. There is no exchange of genetic material. It's kind of creating a clone of itself through a seed. And so we have here just a few examples. What is considered a plant species. Plants that are able to breed with one another will normally be within the same species. Now, species can have different barriers, geographical barriers, that will prevent them from crossing with one another. And that's how they would separate from one another, eventually becoming different individuals. And so we have distance, geographical barriers that will prevent different plants from meeting each other. Now, we as humans bring them into close proximity, but if we're talking about nature, they would never meet each other because one of them is going to be in Asia, the other one is going to be in America. So there's going to be geographical barriers that is going to prevent plants from crossing. There's going to be different ecological, meaning plants that grow in mountains will probably never cross or come across plants that grow in this ocean or close by the sea. So difference in ecological areas, desert versus mountains versus coast versus riparian. So those are different geographical, ecological areas. Temporal, plants that will flower at different time of the year. Some will flower in the spring, others will like to flower in the fall, and so pollen will never meet or cross with one another. And then there's going to be mechanical, where petals will prevent this from happening, uh, and or just barriers from the plant. And or etiological pollination. It has to be the right pollinator, whether it be a hummingbird, whether it be a bee, whether it be a bat. And if something were to happen, then we know that the pollen is not going to cross or move, and it's not going to bring about pollination fertilization. And there's going to be some mechanisms where, if for some reason the pollination occur and the hybridization occur, many plants don't like to produce grotesque individuals. So there is such thing as aborting the embryo. So if the flower detects that it has received the pollen from a different individual or accepted the pollen from a different individual, you can have the abortion of the embryo, and that is going to prevent any further development of that plant. Or in many cases, when you have two different species cross, the hybrid is going to be a sterile individual. And the sterile individual is not going to find a nice pair of the same genetic material. So it will never produce a fruit, and that will kind of stop the cycle right there. Now, we as humans can propagate plants through cuttings and or other asexual means, so we can keep them alive. But if we were to consider them in the wild, if it's a sterile hybrid, it's not going to survive in nature. So that's our other mechanisms that the plants have, or other precautions that the plants will take, actions, I guess, that the plant will take to prevent either hybridization from a weird plant or an unwanted plant. They don't want to create grotesque, ugly plants. We can take some of those embryos and rescue them through the process of tissue culture. And this has brought about some weird combinations that should have never happened in nature or naturally. But there is the process of embryo rescue, where humans can open the flower ahead of time before the mechanisms are developed within the flower. They'll put the pollen from a different plant, completely different species, and allow it to fertilize. And before the mechanisms are set to kill the embryo, they will cut it from the flower, put it in a medium with vitamins and minerals, and grow the plant. As if it was an new individual, the combination of the two different parents will give a very different plant. So this is now humans taking what would have never survived in nature and doing something with it, creating, allowing some of these individuals to survive. But we do have certain specific hybrids that are going to be important. So here's from two different species. So we have citrus, genus Citrus maximum, which is the pomelo, and then that was crossed with the orange, and we have what is known as the grapefruit. So the grapefruit, as we know it, is a hybrid between a pomelo and an orange. And this pink grapefruit, as we know, that's popular here in America, that was developed or hybridized in or selected in Florida, and that's why that tends to be the favorite one here in America. But pomelo grapefruit has been grown in Asia for a very long time. So here is an interspecific hybrid, same genus, different species that give the grapefruit. And then we have the intergeneric hybrid, where we have plants from different genera or different genus. If there's part of the same family, there's a possibility. If they're part of the same genus, there's a good possibility. But the further you get from there, the more difficult it is to get actual hybrids. And so here we have the genus Fatsia, Japanese Aralia, with the genus Hedera, or the English ivy. And when they're able to cross, and then the hybrid is Fashidera, which is a combination of both individuals. It has the traits of both parents. So in certain cases, the crossing of two different genera within the same family, or two different genus within the same family, is possible, as in the case of ivy and Fatsia. And orchids are going to be the ones that are going to be the worst, because they have no mechanism for stopping the pollen from a different orchid species. Their mechanism for preventing selfing or being pollinated by a very different species is going to be their mechanism of pollinators. So that's going to be putting the pollen on different insects, different body parts. And so they rely on that. So they never really develop the genetic barriers. And that is why there are over 100,000 hybrids, orchid hybrids, and you have hybrid after hybrid after hybrid after hybrid that you can keep on crossing and come up with very different colors and different sizes of flowers. So orchids would be the worst. That is just, there was no mechanism, and so they will take the pollen from any other orchid anywhere, anytime. So hybridizing entails the transfer of the pollen from a stamen onto the stigma of a different species, and often taking the pollen from one flower to the other, and vice versa, taking the pollen from the other plant and putting it on the original one, can bring about different crosses or different results. Who is a pollen donor? Who pollen sperm donor? Who's the ovule or the egg donor? Does make a difference as to the possibility of coming up with something new. And so here is just a quick example in the passion flowers. And there's going to be some steps that you may need to take. Obviously, we don't want bees to go in there and bring pollen from an unknown source. And so it may be necessary to either close the flower to prevent insects from going in there, or put a bag on the flower so that it prevents another insect or an insect from bringing the pollen from an unwanted individual, because now you have no control. And so that is kind of the process that leads into the creation of new plants. So every year we have the introduction of new selective varieties, new rose types, new fruit varieties, and they all go through a similar process. It's a very long process because you're going to have to grow different generations and select the traits. So for example, we may have here plants from the wild that we are going to bring and cross it with a domestic type. We want to bring diversity. We want to enhance perhaps the resistance to diseases. Maybe we want to increase the yields. And so we are going to take the flowers and we're going to cross them, and we are going to grow the seeds. We may grow 1,000 plants. And then as they're growing, we can check for vigor. We can check for how robust they're going to be. We can start looking for traits that we like, also heights. And then we are going to grow those out, thinning out some of the ones that are weaklings, thinning out the runts. And then we can cross the offsprings from this and continue on selfing and selecting, selfing and selecting, selfing and selecting until we have the desirable traits that we want. And once that happens, now we can continue on breeding with other plants to bring about resistance if we just got the kernel, the size and the number of seeds and all of that, until we eventually find that one variety that is we're gonna bring out into the trade. And that could be that Gala apple, that could be this Granny Smith and our selected plants. And so it is a very long, tedious cycle of selecting plants, growing them, their offsprings, selecting the best ones, keep crossing them, all done in a controlled environment, hopefully, so that you know what traits are going into what, and hopefully not a bee is not going to contaminate the work. And so we have more or less a cycle of 10 to 20 years, depending on the crop. This would be for something that would be very quick and easy, keeping in mind that if you're doing something like this to an apple, you would have to grow the plant for at least 10 years, that seed, before you realize that it has potential or it does not have potential. So this is what has been happening. So the plants that are being introduced into the trays as new selected varieties in the current year, the work started maybe 10, 20 years ago, and it has taken them this long to develop some new varieties, maybe some new varieties that are going to have good uh, rootstocks for propagation or grafting. Those are also going to be selected. And so tedious process, good for you to understand. And if you ever want to engage in a hybridization project or hybridizing project, then it's going to be good for you to understand this. So with that, I will end. Have a great day. Bye.